from Stony Brook University. And if everything is settled, please go ahead.
Okay. Well, thank you very much. Thanks for Edgar and the organizing committee for giving me this opportunity to share our recent work. So I'm going to talk about some approach that we have been doing for the last few years on dynamical aspect of the non-equilibrium systems. I mean, we heard a lot about the thermodynamic aspect. We are going to concentrate on the dynamical aspect of that. So I'm going to focus first on some theoretical aspect, and then look at particularly two systems, fundamental for the cell function. One is the cell cycle, the other is the cell differentiation and reprogramming. And then talk about some of the experimental quantifications of this driving force. So as we know that, well, as the smallest living entity, cell is very dynamic and is certainly in equilibrium because constantly you receive particle flux, energy flux, information flux coming in and out. And so, for example, this is a illustration of gene regulatory networks, and certainly they receive the signals at the surface and transmit these signals all the way down to the gene to perform the function. And all these processes, like gene regulation, cell signaling, are tightly controlled by the chemical reactions underneath. I mean, so we have lots of molecules interacting with each other. So we can think about the cell as a chemical reaction soup. So while we look at those problems, we can sort of abstract this problem mathematically into the following equation. The x here represents not a space, it's a concentration, for example, or gene expression. And fx is just the chemical reactions of all these molecules within. So we want to understand the general dynamical properties of this. And the typical way of analyzing this is to find the fixed points of this equation and do the linear stability analysis around those fixed points. And that gives you the local stability information. What that doesn't give us is the global information, how you go from one fixed point to the other, how you connect these two, which is very essential in the biology. And so this typically requires some kind of global description in order to connect the local fixed points. And in terms of equilibrium physics, we know that if we know the interactions, we know by Boltzmann law, we know the probability of the state, or the weight of the states in each state space point. So that basically gives you the connection between two local fixed points. So that gives you a global description, and further than that, it's the gradient of which gives you the dynamics, where you go next. And this applies to, I mean, I just give a few examples in biology, like protein folding and protein-protein interactions. This is successfully applied to those. However, when you look at the cell, as I said, it's unlike the folding and the binding, those can be spontaneously occurring. In the cell, as I said, the net flux from the material energy preclude this expression. In other words, the driving force of the dynamics usually cannot be written as a pure gradient per se. And if we can't be written as a pure gradient, so how do we do the global analysis around it? So that's the sort of the problem. So there are a lot of people working on non-equilibrium dynamics, and for the time being, I just discuss our approach. So what we have found, I mean, is the following. So this is still the dynamics. What we have found is although in this kind of open non-equilibrium system, you can't write down the driving force as a pure gradient, but you can write it as a gradient-like force plus something else. And this something else is very important. First of all, this something else is not arbitrary. And second of all, this force is directly related to the net flux coming in or out of the system. And so this is the measure of the non-equilibriumness, if we say, or the degree of detail balance breaking. And plus, this has an interesting feature, it's rotational. And so, so I'll give you a very quick and brief proof of that. So you start with this equation, and you add a little bit noise. I mean, adding noise is, well, noise is everywhere. So, but here is a trick. But you can put the noise equal to 0 at the end, but that doesn't matter. If you put a little noise in it, then the trajectory is stochastic. Then you can't use this to predict what's happening. It's because it's stochastic. What you can predict is the probability pattern, which satisfies this Fokker-Planck equation, whereas this is essentially the probability conservation. Okay, so if I look at the steady state, and so this dp dt equal to 0, we know that this flux is equal to 0. By the way, this flux is determined by these two forces. One is the deterministic force. One is determined by the stochastic force. So you have several possibilities. One is that the flux itself is equal to 0. If the flux itself is equal to 0 and you move this to the other side, you recover the equilibrium situation where the net flux is equal to 0 means detail balance is preserved, and then the force is just a gradient of the potential, and that's basically the equilibrium one. However, the divergence of the flux equal to 0 doesn't necessarily mean that the flux itself has to be 0. So if the flux is not equal to 0, and you move this term to the other side, then you can see that the force has two components. One is similar to the sort of the gradient force that we have seen before, analogy to the equilibrium case. The other one is this the flux and divided by the steady state probability distribution. And notice that this is analogous to the equilibrium, but it's not quite, because here is the minus, the potential is the minus log of the steady state probability, not necessarily the equilibrium probability. So that defines the landscape. This is the net flux in and out, which has the rotational feature. So this is how you do the decompositions. You have two driving forces. One is gradient-like, and the other is rotational-like. So the picture looks like this. In the equilibrium landscape, the dynamics, you just go down, the gradient. But in this non-equilibrium dynamics, you have a spiraling force contributing to the whole. And if we look at the gradient part, the potential part, and we take the noise equal to 0 limit, and one can prove that this function, this gradient part of the scalar potential is a Lyapunov function, which can be used to characterize the global stability of the issue of the system. The other thing is what will be the effect of this additional rotational flux in terms of non-equilibrium dynamics. So you have this kind of landscape, minus log of the steady state probability distribution. And this is, if this is an equilibrium landscape, then with the detail balance preserved, then the best path of going from here to here is to go through the saddle. And going forward and backward paths will be the same. However, if you have that additional rotational force, so the forward path and backward path is not the same. And in general, in the finite noise, it's not going to be passing through the saddle node. So first of all, how you go from one to the other state is changed. And second of all, the rate, you cannot use this naively expected saddle point to determine the barrier height. And you have to use the path-dependent barrier here and here to determine the rate. And so one can see the effect of the two forces. The other aspect we talk quite a bit in this workshop is that the fluctuation dissipation theorem. When you have a detailed balance, there's no doubt the response is equal to the equilibrium fluctuation. But once you have this rotational component of the non-equilibrium driving force, the response and the fluctuations is not quite the equivalent to each other, and there's this additional contribution from this rotational force. The other part is, in terms of thermodynamics, if you have detailed balance, then the entropy production rate is just equal to the negative of the free energy change. However, if the detailed balance is broken, then you have this additional term called the housekeeping term, which is directly related to this rotational force. So there is some kind of basis for the thermodynamics coming from this non-equilibrium driving force. The last one is you can write down the Fokker-Planck diffusion equation into a covariant form. We can write it that way, and the driving force then becomes a gauge field. And this gauge field, when the detail balance is preserved, this gauge field is flat. On the other hand, if you have this rotational force and detail balance breaking, you have a curvature, which is associated with the heat. So, I mean, all these are kind of abstract, so let me go to a more concrete example, which is the cell division. I mean, we know that a cell has several fundamental functions. One is to copy itself, and the other is to differentiate. So those are the two fundamental functions of the cell, which is the smallest unit of the living system. So we know that this cell cycle, is called cell cycle or cell division, is tightly controlled by the underlying gene regulatory networks. We just show an example here, which is the mammalian cell cycle, which has all these genes interacting with each other. The arrows represent the activation, the bars representing the repression. And this forms the cell cycle going through several stages, G1 as G2, and M stage, and going through this cycle. So this is the biological textbook explanation of the cell cycle. The cells start with G0, G1, and going around through checkpoints as G2, and then M phase, and eventually segregated. So from the dynamical perspective, we can lay down, look at this tightly regulated gene networks, write down the corresponding equations, ordinary differential equations, and then start to analyze those. And then we do the splitting of the driving force, decompose the driving force into two. And one is as I said, it's the landscape, the gradient part, the landscape, which is the minus log of the steady state distribution. And this is the vertical axis. The horizontal axis corresponds to the gene expression that we have, two of the gene expressions. And you can see that the landscape looks like an irregular Mexican hat. I mean, basically the center has island height, and the outskirt is high. And in the middle, there's this low potential valley. So everywhere you are here, you get attracted to this valley. And so if this is the only driving force, then you're going to likely to get attracted to the G0, G1 state, and without going through the cycle. So it is this purple line, the rotational force, the presence of the rotational force, which drives the cell once they are in the valley going through the cycle, coherent oscillations. So there are several things which is interesting. First of all, we can identify the few phases that biologists talk about, which is the local minimum along this valley. Okay? And the other thing is you have bumps along this valley, so this corresponds to the checkpoints. And you may wonder, what's the sort of the importance of using this sort of a global pictorial way of understanding the cell cycle? And one of the important things is that cell cycle is a hallmark of cancer. So the cancer cells has a much faster cycling speed than the normal cells. So one could ask what kind of cell which can give you much faster speed and the oscillation than the normal that we have. So, and we notice that this island height and even this along the valley, this local bumps, and also this global flux, these are all globally characterized quantities. So we can ask what are the changes of the underlying structure of the landscape would change this, the global flux, the island height, and this local bump height, because that will influence the speed of the cell cycle and the quality of the cell cycle. Because if this guy is low, you go faster. If the global flux is high, you go faster. And if this island is low, you can easily go from here to here. Then you lose the track. If coming from here or coming from here, you lose the sort of coherence of the cycle. So this is going to be bad. So
question.
Using the
question. I can't read what are the axes there?
The two axes are the two gene expressions, CDK1 and CDK. It's actually here.
And where is that minimum?
Where is the minimum? The minimum is essentially the G1, G0, G0 state. Yeah, is in the cell cycle. Yeah. And so from this, and we can, since these are all global, we can ask the question, which underlying structures are important? So we can identify in this network, these are the key genes we should focus on, and these are the key regulations we should focus on. And so this is the, just go quickly. And another example is the cell not only do divide, but also they differentiate to go from like the primary stem cell to the differentiated cell, like our ear, our liver, or our anything. So a while ago, Waddington has asked his artistic friend to draw a picture like this. So here's the primary stem cell. The differentiation starts and like a marble moving along the valley. So they go down the valley. So this is what Waddington envisioned, the differentiation developmental process. So what we asked is, can we quantify this picture? So we took a very common circuit for the development. There are two genes mutually repressing each other and self-activating each other. I mean, this circuit appeared again and again in the differentiation and development. It's a core circuit, and then this axis represents the sort of the gene expression axis, because we have two, so we actually have a sort of a combination of the two because we run out of the axis, and this is the self-regulation axis, and the vertical axis is the landscape. So what happened is that we can, for this circuit, we can quantify the landscape, by calculating the potential, the steady state probability distribution, and take the minus log of that. And for different strains of the self-regulation, you have different shape of the landscape. So Waddington landscape should be understood as a dynamical landscape. It keeps on changing. The regulation keeps on changing. It's not fixed. And during the development, this regulation changes, and this is what has been found in the experiment too. And so this can quantify the Waddington landscape, where they have this sort of qualitative picture. And one thing important to know is that this is a basin rather than the top, and different from Waddington, and also forward and backward paths are quite different. So in this way, we can quantify the differentiation path, which is the forward path, and also the backward path, which is the reprogramming path for the tissue regeneration. Yeah. We also look at some more realistic networks of the cell differentiation and development, and we found this also this picture of the essentially the landscape of this primary stem cell differentiated to the differentiated cell. We can identify the two paths. One is the corresponding to the differentiation path, which is this one, and the other corresponding to the reprogramming path. So what's important here is we can do the same. Let's see. We can do the same global stability analysis, looking at the barrier, and ask what underlying gene circuit changes could change this a great deal, and find out this global sensitivity analysis, which are the key genes and key regulations in this network. So we identify quite a few. The octal shapes are the important genes, and the solid lines are the important regulations. And one thing that we have found, some of them are consistent with what has already been found in the experiment. But one of them we have found is this PBX1. This is not commonly studied. So an experimental group heard our studies, and they decide to go ahead and look at this particular gene. And what they have found is that, first of all, this particular gene enhanced the stem cell generation. And second of all, this happens to be a therapeutic target for the melanoma. And third is it can rescue the hair follicle cell, mesenchymal stem cell synasis and apoptosis. So they are looking deep into it and try to start a company on that. So we have applied this thing to intracellular network and intercellular networks, and also some of the physical systems, and it has been summarized in a few reviews. Now, lastly, I want to talk about, we talk about the importance of the landscape and the rotational flux component of the driving force. Can we experimentally quantify those? And so we decided to collaborate with the experimentalists and look at some of the biological system to see if we can do that. The first one we have tried is the so-called hydrogen atom in molecular biology, is the bacterial lambda-phage, where typically you have this two genes mutually repressing each other. It's supposed to be like a relatively simple one. So, so here's the technique that they used to study this problem. Since you want to study the dynamics, and the typical way of doing that is to use fluorescence to tag those molecules and see what happens. But since you have two genes, and people typically use two different kind of labels on it, but then you have technical issues, like their maturation times are different, so they can't coincide with each other exactly what what happened here is not necessarily exactly what happened here because they have different properties. So the technique here is to use one fluorescence label rather than two. So one is, but at the different locations of the cell. One is called Venus. You sort of localize around the cell surface, and the other is localized within the cell, roughly in the center. And so using these two probes, we can do a temperature mutated sample. And what happened is that you can see at a certain temperature, lower temperature, you only see one kind of genes. And if you look at higher temperature, you see another kind of genes. And it's in the middle, you can see both simultaneously in different locations. So in the microscopy, you can monitor these trajectories, the real-time trajectories of these two genes. And this shows that 4 different colors, the 4 different colors corresponding to 4 different states. And for example, this green represents the C1 is dominating. C1 is bright. And this purple, this purple one represents both are high expression, and this blue one corresponding one is low. The crow is low. And then you can do the statistics of these trajectories. And instead of expected 1001 states, you actually find 2 more states, which corresponds to the binding completely to the binding side or the completely loose side. And that's something that we have found. Instead of 2 states, naively expected 2 states, there are 4 states. And because of real time, we can do this hidden Markov chain analysis and look at the transition times, et cetera. One thing which is interesting is that we found that the state 10 and 01, they don't directly go switching from one to the other. This takes forever. Instead, they go through this bridge of 1 state to the 10 state. And this is also not expected before. But this histogram gives you the landscape, so we can quantify the landscape. On the other hand, we also look at what about the other component of the driving force. We look at, for this, we look at a single molecule enzymatic reaction dynamics. And as we know that for the enzymatic dynamics usually follows this Michaelis-Menten rule, where the inverse of the rate of the enzymatic reactions is inversely related to the substrate concentration. This is for 1. So the example that we have the horseradish peroxide catalyze the 2 substrates instead of 1 substrate. And there's an interesting property we can sort of simplify into this picture. So here's the enzyme, which has 2 conformations. And here's the catalyze the one reaction. This is the catalyze the other reaction. So from this graph, one can show that if there's no internal flux going on, if the internal flux is 0, then we go back to the famous Michaelis-Menten single molecule reaction rates. However, if there's any net flux flowing around here, then there's an extra term into this so-called Michaelis-Menten type of rate equation. So it's not Michaelis-Menten-like. So while this is a relatively simplified picture, so we can connect this to the observations through the correlation function. And then this is the single molecule experiment, this is the enzyme, this is the substrate, and this is the product. And taking care of all the diffusion effects, and here's the result. Interesting enough, this is the inverse of the rate and the inverse of the substrate concentration. If it's Michaelis-Menten, it will be straight line. And as one can see, this is not a straight line, it's actually curved. And this is immediately saying it's very much deviated from the Michaelis-Menten relationship or the Michaelis-Menten rate. And that's because this internal flux is not equal to 0. And there is a dependence of internal flux versus the substrate concentration. And from this, we can also calculate the chemical potential, which is roughly a constant at a specific substrate concentrations. We can also, from the experimental data, we can calculate the entropy production versus the substrate concentrations. And it's also, we can show that it's related, this flux is related to the time reversal symmetry breaking. One last thing is that this is all low throughput experiment, like a few molecules at a time. Then most recently, people have developed a technique for a single cell RNA-seq, so can simultaneously monitor many genes at the same time, but not at a real time, but basically at some time slice. So one could go ahead, I'm not going to bother with the detail, but one can go with the data and find out, since we know the data and we can find out the V x dot is, we know what the force is and fit with the force. We can fit this vector field, and then you can, from there, you can decompose this, from the data, the driving force in terms of landscape and the flux. And this could be just directly coming from the experimental data. And for the differentiation also. Let me thank my collaborators in Stony Brook, many of my graduate students and postdocs, and also the several experimental work that we collaborated with Johns Hopkins University and the Chinese Academy of Sciences. Thank you.